onorată asistență, distinse domnule academician James Edward Rothman, onorate domnule președinte al Academiei de Științe a Republicii Moldova, domnul Ion Tighineanu, distinși oaspeți, înalt senat, dragi colegi, stimați studenți, onorată asistență. Secole la rând, în învățământul universitar mondial, dăinuie o frumoasă tradiție de a acorda titlul onorific de Doctor Honoris Causa celor mai remarcabile personalități la nivel național și internațional, în semn de profundă gratitudine și înaltă apreciere a meritelor deosebite și contribuții substanțiale pentru dezvoltarea societății și progresul efectiv al omenirii. Cu sprijinul și ajutorul și participarea nemijlocită a domniilor lor, universitatea se alinează la standardele internaționale și exigențele contemporane. Se înregistrează în sistemul mondial al învățământului superior medical și farmaceutic și în area europeană a cercetării și pentru universitate este un mare prestigiu. Se creează condiții favorabile pentru realizarea mobilităților academice, atât a studenților, cât a profesorilor, personalul științific, didactic. Se deschid noi perspective reale de promovare a imaginii instituției noastre pe arena mondială. Ședința festivă de astăzi, în conformitate cu decizia Senatului din 26.08.25, proces verbal numărul 6/15, este consacrată conferirea titlului onorific de Doctor Honoris Causa al Universității de Stat de Medicină și Farmacie Nicolae Testemițanu din Republica Moldova, domnului James Edward Rothman, remarcabil cercetător cu renume mondial, laureat al Premiului Nobel în medicină în anul 2013, profesor emerit Sterling de biologie celulară la Universitatea Yale, Statele Unite ale Americii. Domnia sa astăzi ne-a onorat cu prezența. Dați să-l aplaudăm. Dumnealui este însoțit și de doamna, soția dumnealui, care este tot profesor universitar. Dați să o salutăm. La ședință participă șef de catedre subdiviziuni, profesori, studenți, medici rezidenți, studenți doctoranți, reprezentanța ai Academiei de Științe Republicii Moldova, academicieni. Deci, și sigur că membrii Senatului. Distins Senat, dragi colegi, onorată asistență, permiteți să declar deschisă ședința festivă. Mulțumim. Onorată asistență, prezint atenției domniilor voastre laudațio în onoarea distinsului academician James Edward Rothman, cu ocazia acordării titlului onorific de Doctor Honoris Causa al Universității de Stat de Medicină și Farmacie Nicolae Testemițanu. Profesorul Rothman este recunoscut ca unul dintre fondatorii biologiei celulare moderne. Domnia sa are contribuții fundamentale la elucidarea mecanismelor moleculare ale transplantului intracelular prin vezicule și la formularea ipotezei SNEA. Această ipoteză sau termen desemnează o familie de proteine esențiale pentru transportul intracelular prin vezicule, adică pentru procesul prin care celulele mută substanțele dintr-un compartiment în altul. Acest lucru a revoluționat înțelegerea proceselor de secreție celulară și de transmitere a semnalelor nervoase. Pentru aceste realizări, domnia sa a fost distins cu numeroase premii internaționale, printre care premiul Lasker pentru cercetare fundamentală 2002, premiul Kevli pentru neuroștiință 2010, premiul Nobel pentru fiziologie și medicină 2013. În cariera sa academică, domnul James Rothman a îndeplinit funcții responsabile la instituții prestigioase cu renume mondial. Profesor la Universitatea Stanford, 1978-1988. Profesor la Universitatea Princeton, 1988-1991. Șef al programului de biochimie celulară și biologie moleculară la Centrul Oncologic Memorial Sloan Kettering, 1991-2004. Profesor și director la Universitatea Columbia, 2004-2008. Iar din 2008 la Universitatea Yale, unde a condus Departamentul de Biologie Celulară până în 2023 și deține titlul de Sterling Professor. De asemenea, a executat și exercitat funcții responsabile de director a Institutului de Nanobiologie din Yale. Profesor asociat la Colegiul Universitar din Londra și la Școala Normală Superioară din Paris. Profesorul Rothman este membru al celor mai prestigioase academii și organizații științifice, printre care Academia Națională de Știință, Statele Unite ale Americii, Institutul de Medicină, Statele Unite ale Americii, Societatea Regală, Marea Britanie, Academia Europeană, Organizația Europeană de Biologie Moleculară. Contribuțiile sale remarcabile includ participarea în comitetele editoriale ale unei reviste științifice de prestigiu. De asemenea, este membru în juriul premiului Leiscă Award. Activitatea didactică și științifică a remarcabilului profesor este reflectată în peste 300 de articole publicate în reviste de top și în conducerea unor centre de cercetare de excelență, precum Departamentul de Biologie Celulară și Institutul de Nanobiologie de la Universitatea Yale. Domnia sa a format zeci de generații de cercetători și a contribuit decisiv la consolidarea școlii științifice în biologie celulară și medicina moleculară. Recunoașterea aportului considerabil al domnului profesor Rothman prin acordarea titlului de Doctor Honoris Causa a USMF Nicolae Testemițanu constituie un gest de înaltă gratitudine și apreciere, consolidând în același timp vizibilitatea internațională a universității noastre, a academiei noastre și a țării noastre. Înalt Senat, distinși oaspeți, dragi colegi, studenți, din cele relatate mai sus, constatăm cu satisfacție că prin activitatea multilaterală, prodigioasă, prin sprijinul și cu ajutorul acordat sistemului de sănătate mondial, prin descoperirile fundamentale în domeniul biologiei moleculare, ce constituie temelia tuturor proceselor biologice normale și patologice ale organismelor vii, inclusiv a oamenilor, academicianul James Edward Rothman merită titlul onorific de Doctor Honoris Causa a Universității de Stat de Medicină și Farmacie Nicolae Testemițanu din Republica Moldova. Aplauzi, vă rog! cuvânt în susținerea domnului profesor James Edward Rothman se oferă domnului academician Ion Tighineanu, președinte al Academiei de Științe a Moldovei. Vă rog, domnul academician.
Stimate domnule rector, distinse profesor James Rothman, distinși cercetători științifici, onorată asistență, bună ziua! Vreau să menționez că avem la Chișinău, în Republica Moldova, zilele acestea, un oaspete deosebit. Profesorul James Rothman, laureat al Premiului Nobel pentru Fiziologie sau Medicină, este una dintre cele mai respectate figuri ale biomedicinii moderne. Cercetările sale au revoluționat înțelegerea mecanismelor fundamentale prin care celulele comunică între ele, un proces esențial pentru viață. Descoperirile sale privind sistemul de transport vezicular, intracelular, au explicat modul în care moleculele vitale sunt transmise în interiorul celulei, de la un compartiment la altul, cu mare precizie. Acest dans molecular, cum îl numesc unii biologi, permite secreția hormonilor, neurotransmițătorilor și altor substanțe care mențin echilibrul și funcționarea organismului uman. Născut la New Haven, Connecticut, la 3 noiembrie 1950, 75 de ani în urmă, James Rothman a fost atras de știință încă din adolescență, fiind fascinat de complexitatea vieții. Studiile la Universitatea Yale și la Harvard l-au condus spre biochimie, iar perseverența, gândirea sistemică și pasiunea pentru ordine l-au făcut să descifreze unul dintre cele mai complicate procese ale naturii. Astăzi dimineață, domnul profesor James Rothman a avut o prelegere așa de rezonanță, foarte și foarte interesantă în cadrul conferinței internaționale în domeniul nanotehnologiilor și ingineriei biomedicale. Eu pot să dau citire genericului: bazele biofizice de transmitere rapidă a informației în creierul omului. Este o temă de mare interes și mare importanță. Este semnificativ faptul că profesorul Rothman are rădăcini basarabene. Străbunicii săi emigrând din Fălești și Telenești în Statele Unite ale Americii la începutul secolului al XX-lea. După mai mult de 100 de ani, dumnealui este primul reprezentant al familiei care revine la baștina strămoșilor săi. Un moment de profunzime simbolică în care știința și istoria se întâlnesc. Astăzi, ca profesor la Universitatea Yale, James Rothman continuă să inspire generații de cercetători prin exemplul său de rigoare, modestie și viziune. Prin munca sa, el ne amintește că adevărata cunoaștere nu constă doar în descoperiri legile naturii, ci și în a înțelege ordinea și frumusețea ascunsă în fiecare celulă a vieții. Vreau să mai adaug că domnul profesor James Rothman l-a cunoscut și au colaborat cu profesorul George Emil Palade, care s-a născut și a copilărit și a avut studii la Iași, a plecat în Statele Unite și a devenit un mare om de știință, considerat astăzi părintele biologiei celulare. Și domnul profesor Rothman, cu timpul, a devenit director al Institutului, care a fost fondat cândva de profesorul George Emil Palade și a dus cu cinste mai departe activitatea științifică, devenind în 2013 laureat al premiului Nobel, tot așa cum a fost și George Emil Palade. Vă mulțumesc pentru atenție.
Onorate domnule profesor, James Rothman, vă rog, treceți lângă mine, please.
Please go. Yeah, you can take. Yes.
În numele Senatului și a comunității universitare, primească atribuțiile asistență, îmânesc domnului profesor diploma de Doctor Honoris Causa al Universității de Stat de Medicină și Farmacie, Nicolae, Testemițanu.
Thank you. I'm hoping everyone speaks English a little. Unfortunately, I cannot address you in your language. Thank you. This is a tremendous honor for me, and I genuinely am moved by all of you being here. Thank you. Very good. Yesterday was a very interesting experience for me. As was mentioned, my grandfather came from Felești. My grandmother came from Telenești. We'll be visiting Telenești on Saturday. Yesterday, we had the experience of walking on the market street in Felești over stones that were clearly from the 19th century, and I felt like I had come home. And my wife, Joy, and I have felt so welcomed here in Chișinău and also at the Academy of Sciences. And we so appreciate the honor that this represents at many levels. The name George Palade was mentioned. already. He was an inspiration to me. I knew him, obviously. I'd like to mention the name of another scientist, Professor Randy Schekman. He and I shared the Nobel Prize. And I think in one of the most remarkable stories in scientific history, his family also comes from Bessarabia. What are the chances that two scientists would would end up getting the Nobel Prize from Bessarabia? What are the chances that those two scientists would be working on the same problem? What are the chances they would share the same Nobel Prize? What are the chances that the problem they were working on was based on the earlier work and discoveries of the famous Romanian scientist, George Palade? Almost zero. That is why it is an exceptional story for all of us, including you. And so this entire occasion is very special for me, as it is for the field of cell biology. Now, I was asked to tell you a little bit about my history and how I came to be here today. And I'll try to do that in a modest way that also might be interesting, hopefully, to you. Okay? So I have a couple of slides, and I'm hoping that the technology will work. So, could I have the next slide? I don't know how to change the slides. Okay. Do I just touch it? Yeah. This is more high tech, I have to tell you, than any. Yeah. You have a fantastic new building here. We did have the slides before, you know?
Yeah, 1 second.
Yeah, I mean, before we got the people involved, it worked really well. Huh? Yep, we're back. But how do I change the slide without losing it? Perhaps this is the answer here?
Yeah, now it's working. Yeah.
This one?
Pointer, yeah.
Good. I was born in the year 1950. I grew up in a small village near Boston, Massachusetts, not too far from New York, where my father moved from New York City, where they emigrated, as you heard, in around 1910. The era I grew up in is sort of illustrated by this picture. It was the Cold War. Those of you who may be my age were in Moldova were on the other side of the Cold War. What we had in common was something called the space race. And the Soviets, when they developed the first step in the space race, they launched the satellite Sputnik, very famously in 1957. And that brought about an enormous response in the United States, one of fear. And the fear was that unless we, the Americans, had a position in space, that we would become victims of nuclear bombs dropped from space. Of course, that was a fiction, but so many things in life that derive politics are actually fictions. But it was a useful fiction in this case because it galvanized American interest in science. And for the last 60 or 70 years, the United States has been a powerhouse in science, initially in physics. And as a result, in this era, when I was very young, the physicists were the heroes in the world. People like Einstein and Oppenheimer, one of the first astronauts that you see here, Alan Shepard. And of course, we lived in fear of what you see over there, which unfortunately we still do, which is the weaponry associated with nuclear power. Well, I then was wanted to be a physicist. I knew I wanted to be a scientist, and physics is what attracted me, probably because there was so much interest, for the reasons I just mentioned, in the field of physics. Here's me at the age of 12. Hopefully you could still recognize which one is me. At the age of 12, building a rocket. And from that rocket, which did not go up to a great height, but it did go, I had to figure out how high it went. And so I taught myself trigonometry. And then I taught myself geometry. I had to learn how to ignite the rocket. So I had to learn how to build electric circuits. So I taught myself electromagnetism. So by the time I was ready to go to university, and I went to the university at Yale, that's where I went to college. These are my parents who are dropping me off in 1967 in my first days at college. From that time, I wanted to be a physicist. And I studied physics at Yale. That still influences the way I think about the world. But my father, who you see over there, was a physician. He became a doctor in the United States. His father was born in Filesht and lived there until he was about 25. So there's a direct connection there. And my father said very wisely, you really ought to study some biology because actually, you know, there aren't that many jobs for physicists and maybe you should be a doctor. So I started studying biology in my last year in university, and I fell in love with it. And I dropped physics very quickly. It never left my brain, but I stopped working on it. I then went to medical school to become a doctor. That didn't last very long. It lasted about 2 years, and I left medical school to go into research, which was always my passion. But while I was a medical student, studying, maybe some of you are medical students. Are there any medical students here? I hope so. And who is a medical student? Could you raise your arm or hand? Ah, very good. When I was in my first year of medical school, I heard a lecture about this gentleman's research. It was very new then. It had barely been published. This is Professor George Pilati. He discovered many things. He was perhaps the first scientist to use the electron microscope to look inside of a cell. And so as a result, he was privileged to see things that no human being had ever seen before. And one of the things that he discovered is that there are a series of organelles in the cell, which he named, like the endoplasmic reticulum, the Golgi apparatus, named after the famous Italian physiologist Camillo Golgi, and other organelles. And Pilati discovered a process by which little vesicles, which he calls here in this diagram from his Nobel Prize lecture in 1974, he calls this a Golgi vesicle. We today call it a Pilati vesicle, but he was very modest. And these vesicles are very tiny. A typical cell might have 100,000 of them. And they swarm around the cell carrying cargo from one place to another as is necessary for the cellular economy. And I won't go into that in detail, but Pilati discovered that this happens. What he could not discover, because the technology was not available to him, was how these vesicles form, how each one knows in the maze of cellular membranes where to go, and how that vesicle knows how to deliver its cargo by a process that he named called membrane fusion. None of the molecular mechanisms could be uncovered because the technology and the methods did not exist in his time. Now, I, on the other hand, grew up much later than he did, and I was trained as a biochemist, not a cell biologist. And as a biochemist, I knew that the way to uncover molecular mechanisms was to try to reproduce them in a test tube system and to treat it as if it were an enzyme assay, to try to recapitulate outside of the cell what goes on naturally inside of the cell, and transforming it into a chemical system with which chemical questions can be asked and chemical answers sought. And so I made it my mission, actually, even as a medical student, to try to understand this problem. I was so inspired. I could not imagine how these vesicles would form from other membranes, how they would, without any human intervention, know where to go, okay? And I thought there are really two hypotheses. There were two hypotheses to try to explain this process. And when I moved, as you heard, to Stanford University to start my own laboratory, I came back to the problem that Pilati had introduced, but now with modern biochemical methods trying to solve it. And I wanted to see if I could put all of this complexity on the basis of simple chemical mechanisms. Ultimately, my colleagues and I succeeded, but it was the work of almost 20 years. I will try to summarize in about 5 minutes those 20 years. But briefly, there are two ideas that, I don't have a pointer, but that's okay. There are two ideas that contrast with each other. One idea is what I call anatomy dictates specificity. This was the prevailing idea, and actually it was Professor Pilati's idea. It turned out not to be correct, which illustrates, by the way, a very important point. Nobel laureates can be wrong. I've certainly been wrong many more times than I've been right. The important thing is ultimately to be right and to be open to criticism and to new ideas, as Pilati was to mine. And what Pilati and others thought was that the cell is so beautifully organized that a vesicle coming from, for example, the endoplasmic reticulum could find its way to the Golgi, the saccules that you see in the center, merely because the Golgi is there. What I thought was that the other possibility was that the vesicles have a built-in chemical specificity. You could introduce the vesicle anywhere in the cell, and it would find its way to the right location, based on its ability to bind to the correct target when it got there. The Pilati idea, the conventional idea, did not lend itself to any studies outside of the cell. Because if you think about it, if the organization in the cell is broken when you homogenize a cell to release its contents, which a biochemist must do, if you do that, if the organization in the cell is necessary, then the process will no longer occur. But on the other hand, if the vesicle can figure out where to go based on its intrinsic specificity, there's at least hope to be able to reproduce it outside of a living cell. So the test of the hypothesis, really, is to see if the process of transport by vesicles between two locations can occur outside of a cell. And we discovered that that, in fact, could take place in the late 1970s. And I won't reiterate the experiment that is involved, but briefly what it represents is mixing extracts from two cells. We took one population of cells on the left, a second population of cells on the right. We broke each of the cell populations, and we mixed them as soluble extracts. And what we discovered is that proteins initially resident in the membranes of one cell could move correctly to the correct locations in the other cell, even though the cell wasn't there, even though the organelles were just floating around. That was an amazing discovery. It took many years for my colleagues actually to accept it. It took 15 years, really, for this to be accepted as truth. But ultimately, it was accepted. And the result of that is that we were able to, okay, there. We were able to utilize what is now called a cell-free system. We could take one set of membranes and incubate them in the test tube with the energy source ATP, and they would produce the transport vesicles. So up until this experiment in 1989, any vesicle that you ever saw could only be seen in an electron microscope image after the cell was dead and fixed. I hope I'm being clear. These vesicles are the same transport vesicles, but they are now produced in the test tube. And we were able to purify them from the extract that produced them. So this is a test tube full of pure living transport vesicles. That means they can still function. So now these vesicles can be added back, again in the test tube, to in a second reaction to the membranes with which they should fuse. And indeed they do fuse, and that's how we know that they're functionally competent. We were able to characterize the proteins that surround them and characterize completely as a chemical reaction the mechanism by which they bud from the membrane. When we had these vesicles in hand and when we mixed them with the target membrane, which in this case was the Golgi apparatus, we observed that they fused. But we observed that they would not fuse unless two things were provided besides the membranes themselves. We had to provide a source of energy, which biochemists in the audience will know is ATP. And we had to provide a source of additional enzymes. So we had to add back the cell sap, or the cytoplasm, in order for the vesicles to do their job and wake up and think that they were in a living cell, but of course they were not, okay? My job was to convince them that they were, and we did. And that cell extract had hundreds and hundreds of different proteins in it. Which protein or proteins might be necessary for the vesicle to fuse? Probably not all of them. Well, over a 5-year period, we sifted through all the proteins in those extracts until we found the 2, and there were only 2, that were necessary for these vesicles to fuse. Their names don't matter, but we called them NSF and SNAP protein. The SNAP protein turns out to bind to the membrane where the fusion is going to occur, and the NSF molecule hydrolyzes ATP to power the fusion process. But how does the fusion process occur? And how does each vesicle know to fuse with the correct target membrane, not the incorrect? That answer came when we isolated the receptors for the SNAP protein, which were present in the membranes, and each type of membrane had its own type of SNAP receptor. SNAP receptor abbreviates SNARE. S-N-A-R-E stands for SNAP receptors. That's where the name came from. And in 1993, we isolated the first SNAP receptor, or SNARE. That was done by taking the pure NSF and the pure SNAP proteins, mixing them with the membranes, extracting complexes from the membranes, and purifying them. And for those of you who may have a background in biochemistry, if you look on the far side, you will see what we call an SDS polyacrylamide gel. It separates the proteins into individual molecular species. Each of the dark bands that you see corresponds to a protein. And you'll see they have labels on them. Now, at the beginning of our experiment, NSF and SNAP, alpha SNAP, were the proteins with the labels. We put those in to go fishing. But what we pulled out, in addition, were other proteins, and they did not come with labels. We did not know what they were, but we established through their amino acid sequence their identity. And they are called syntaxins, SNAP25, and VAMP. And their names don't matter, but what does matter is that one of them was in the transport vesicle, VAMP, and the other 2 were in the plasma membrane. And those are the SNARE proteins. And through this experiment in 1993, we discovered what we call the SNARE complex. That is a complex of proteins of which one is in the transport vesicle and the other 2 are in the target membrane, and they partner each other by binding each other to tell the vesicle to go to the right place. And then this gave us the idea, which we called the snare hypothesis, and you heard that term before, that every vesicle in the cell has its own version of the snare protein. Every target membrane has its own version. And we propose that there are two types of snares. One, which is symbolized by the V's that you see, like the yellow V or the green V at the cell surface. And the other set of snares in the target membranes are called T snares. T stands for target. And the idea is, and this is now established as a fact, that the V snares and the T snares bind each other specifically. And so there are many V snares, there are many T snares, but they form specific partnerships with each other. And when they do, the vesicles dock to each other. And as it turns out, in the most beautiful outcome that you can imagine, when the V snare binds the T snare, and I won't show you this, the two actually join the membranes together and fuse them. So the actual fusion machinery are the snare proteins themselves. So these proteins encode in their physical chemistry the specificity of the compartments of the cell that Palade had discovered many years earlier. And if you want to think about it that way, and I think it would be fair, you can say that the specificity of the cytoplasm, how each organelle retains its identity, is encoded in this family of proteins. And the family of proteins has many functions in biology. It is responsible for the secretion of hormones like insulin. It's responsible for the release of neurotransmitters in the brain. It's responsible for the propagation of organelles in the cytoplasm and the plasma membrane itself. And so, in a very broad sense, the existence of a cell and the propagation of a cell, which is, of course, the very definition of what a cell is, relies on these proteins in a fundamental way. Imbalances in the pathways that these snare proteins catalyze show up in diseases. They actually underlie many immunological disorders, numerous cancers, as well as endocrine and metabolic disorders. And I can also go on more recently to include psychiatric disorders. So I'd like to leave you with a couple of thoughts. First, nothing happens on its own in science. I've been privileged over the years to have trained and worked with and learned from over 300 young scientists, many of whom are now famous scientists and department chairs, members of leading academies, and have made discoveries entirely on their own. And so I have enormous gratitude to the many dozens of contributors who've helped me have a lot of fun studying bubbles, because I tend to think of these little vesicles as tiny bubbles in the cell. And then I'd like to finally leave you with one last thought, which is, after years and years of doing science, and now in the latter part of my life, I can tell you the following. When I was young, I used to think I was very clever, okay? Perhaps not some of you students think that you're very clever, and you probably are, and I was, okay? But I didn't realize that as clever as I might have been, or as you may be, how insignificant we are as individuals as compared to the scientific method and the process that we take advantage of. And by the way, this is true whether you're practicing medicine as a doctor, because your success as a clinician is hypothesis dependent. You make a hypothesis about what may be wrong with your patient, and then you test that hypothesis in as many objective ways as you can. It's also true in science. The greatest discovery ever made by science, in science, is and always will be the scientific method itself. If you simply follow the scientific method in your research, in your medical practice, and I would actually argue in life, as a way of trying to maintain objectivity, focus on evidence, you will be well guided in life, and the world will be a better place. Thank you.
Îi mulțumim domnului academician, domnului Doctor Honoris Causa al Universității noastre pentru această prezentare. Sper că de folos și pentru tineretul studios. E ceva legendar, e ceva deosebit, e ceva ce noi toți deja învățăm la biologie moleculară, la biochimie și iată în fața dumneavoastră sunt persoanele care au descoperit acest lucru și vedeți persoanele care au dus faima și au dus viitorul medicinei, deci ca să înțelegem mai bine toate aceste procese, să le cunoaștem, ca să le învățăm și mai mult ca atât sunt deci personalități notorii și sunt descendenți ai țărișoarei noastre, deci atât George Emil Parade cât și domnul academician. Deci de ce pentru noi este o mare, mare onoare și o mare, mare cinste. Dați să-l felicităm încă o dată, să-i dorim mult sănătate. Onorată asistență, cu aceasta ședința...
Domnul rector, se poate doar un anunț.
Sigur.
Da, onorată asistență, utilizez această ocazie să vă aduc la cunoștință că mâine, la ora 14, domnul profesor James Rothman va avea o întâlnire cu tineri într-o abordare largă. Vor fi și elevi care au fost selectați din licee, vor fi și studenți, tineri cercetători și va fi o discuție unul la unul cu tinerii. Și cine dorește să fie mâine prezent la ora 2 în sala Azuriei Academiei de Științe, cu plăcere vă invităm și mâine doamna profesor Joy Hirsch, care este prezent aici, soția domnului profesor Rothman, va avea o prelegere la Academia de Științe și pe site-ul Academiei este informația, deschideți asm.md și o să fiți bineveniți și eu cred că mâine va continua acea mare sărbătoare care a fost generată de vizita domnului profesor James Rothman. Vă mulțumesc!
Noi încă o dată reiterăm din partea comunității academice, din partea a comunității medicale, din partea comunității deci, tineretului studios, să-l felicităm cu acest titlu, să spunem că noi suntem onorați că dumnealui deține acest titlu de Doctor Honoris Causa al Universității Nicolae Testemițanu din Republica Moldova, suntem deschiși prin colaborare, suntem deschiși pentru a avea relații de conlucrare cu dumnealui, cu discipolii dumnealui și deci și ori de câte ori, dacă dumnealui să dorească să vină în Republica Moldova, noi suntem gata să-l primim. Deci. Felicitări, vă mulțumim că ați fost alături de noi astăzi și vă dorim o zi frumoasă în continuare. Mulțumesc!